Hello everybody. Welcome to YouTube channel Enrich Your Biology. I'm here Harpreet Kaur Kapoor to enhance your knowledge in biology from the grassroots level. So in the previous video I discussed about the living and the non-living beings. What are their differences? And today we will do the structure of cell, which is the basic unit of life. Cell was first of all discovered by Robert Hooke, who observed the piece of cork under the self-made microscope and saw some honeycomb-like structures, saw some rooms-like structure, and he called it cell. It is the Latin word cell meaning room. Then it was further studied by A.V. Leeuwenhoek. He observed unicellular freshwater organism under the compound microscope and the higher power microscope. But he saw that it is motile as well. Then nucleus was discovered by Robert Brown. After the discovery of cell theory was put forward by Schleiden and Schwann and further elaborated by Robert Virchow. According to cell theory, cell is the basic unit of life. All the living organisms, they are formed of cell, whether they are unicellular, multicellular, plants or animals. And the new cells arise from the pre-existing cells. So today we shall do the structure of animal cell. Basically the structure of all the cell is the same with few differences according to their functions. How there is some difference between the, very minor difference between the plant and the animal cell. Today we will do the structure of animal cell. Cells are basically of two types, prokaryotic cell and eukaryotic cell. Prokaryotic cells, they are primitive cells. They are not advanced cells. They consist of cell membrane enclosing the cytoplasm, but no nucleus. Rather, the nucleus of dark mass is found in the center, and subcellular organelles, they are also absent. They are very small in size, ranging from 0.1 mu micron to 10 mu micron. Example is bacteria and cyanobacteria. In prokaryotic cell, there is no sexual reproduction. There is only asexual reproduction. They divide by mitosis. However, in some bacteria, conjugation occurs, which is called as this reproduction, but it's not the reproduction actually. They divide by binary fission. So, no, not by mitosis, they divide by binary fission. Eukaryotic cells, they are highly advanced cells. So today we will describe the eukaryotic animal cell. Cell basically consists of three parts: cell membrane, cytoplasm, and nucleus. I am showing the diagram. This outermost layer is the cell membrane. This yellow color which I have drawn, it is clear substance which is called the cytoplasm. And near to the center, there is this purple color which I have drawn. It is nucleus. So basically it has three parts. So we will describe one by one. First of all, cell membrane. This cell membrane actually is it is formed of phospholipid bilayer, two layers of the phospholipid molecules embedded with proteins. The cell membrane, it is selectively permeable. That is, it allows some substances to enter and to move outside, but does not allow the other substances to enter and to move outside. So that's why it is called as selectively permeable, or it is also called as semi-permeable. So in the cell, membrane provides shape to the cell. It separates the cell from the extracellular material, that is matrix, and ultimately the group of cells that parachute to the body of the organism. Then cell is the site of many metabolic activities. So, how the material pass through the cell membrane? These water and gases, they are freely moved inside the cell and out to the outside, from the outside to the inside through the cell membrane. Because cell membrane is permeable to these water and gases. They follow the law of concentration gradient. That is, the substance move from the higher concentration to the lower concentration. So, water enters the cell through diffusion, through osmosis. Water from the inside the cell will move to the outside if the concentration of water inside the cell is more than that of the outside solution. And if the concentration of water is more outside the cell, it will move inside the cell. So you can take the example of an egg. Egg itself is a cell. You know, ostrich egg is single cell. It is the largest cell. It is 5 inches in diameter. So if you remove the outer shell, hard shell of the cell by putting the hydrochloric acid on it, which is formed of calcium carbonate, that will be dissolved and you will see the very thin membrane enclosing the inner mass. So you remove the outer layer, calcium carbonate of the shell, by putting on it hydrochloric acid and take out the egg and put it into the water. What will happen? Water will enter inside the egg and egg will swell up. It will enter until unless the concentration of water and inside and outside is the same. That is the solution, they become isotonic. They simply follow the process of osmosis. If you put that egg into some concentrated solution of sugar or salt, then you will see water will move from inside the cell to the outside through osmosis and egg will shrink. So it means the outside solution is hypertonic than the inside solution. So you know what is isotonic, hypertonic and hypotonic, you might have read in chemistry. Isotonic solution when the solutions are of the same strength inside and the outside. Hypertonic solution when the outer solution is dilute than the inside cell. That is called as hypertonic. And if the solution outside the cell is more concentrated than the cell itself, then it is called as the hypertonic. Similarly, gases, they move through diffusion. They also follow the concentration gradient. If the concentration of carbon dioxide inside the cell is more than that of the outside the cell, that is in the matrix, the carbon dioxide will move outside and the oxygen will move inside. If the concentration of oxygen is more inside the cell than the outside the cell, then oxygen will move outside and carbon dioxide inside. So they follow simply law of diffusion. But other substances, other minerals, you can say substances, they are moved through the cell membrane, to say by taking some energy, and that is called as the active absorption. And some of the substances, they are allowed to move, others they are not allowed to move in and outside. That's why the cell membrane is called as the selectively permeable membrane. So what's the function? It proceeds to the cell. It separates cell itself from the matrix. It is a size, to say, it controls the movement of inside and outside of substances. Then next is inside the cell membrane is enclosed a fluid mass. This yellow color fluid mass is called a cytoplasm. Cytoplasm is clear fluid-like substance. In it are suspended many small minuscules, many small things which are called as cellular organelles, subcellular organelle. There are approximately 7 types of subcellular organelles, which are those. Number 1, this blue one which I have drawn, endoplasmic reticulum. Number 2, these orange color, they are mitochondria. Then in the center is nucleus. Near to the nucleus, here are sac-like structure which are called as the Golgi bodies. Then there are also present some tubules-like structure which is called as the microtubules. These spherical structures are the lysosomes, and these folded structures, they are the centrioles, and some vacuoles are also present and some vesicles are also present. So we will describe one by one all the subcellular organelles. First of all, we will do the endoplasmic reticulum. This blue structure, this one, it is endoplasmic reticulum. It is present near about the nucleus spread throughout the cytoplasm, and it is of two types. Rough endoplasmic reticulum, and this is rough, and smooth endoplasmic reticulum, this is smooth. As the name exhibits, rough endoplasmic reticulum, they are rough in appearance. Why they are rough in appearance? Because ribosomes are present on their surface. On their outer surface, ribosomes are present. Actually, they are membranous tube-like structure. These endoplasmic reticulum, they are a membranous tube-like structure, and ribosomes are present on their surface. The function of rough endoplasmic reticulum is to synthesize protein. Ribosomes, they are the site of synthesis. The function of these smooth endoplasmic reticulum, they are smooth in surface. They are smaller in number, they are present near the rough endoplasmic reticulum. Their function is to synthesize lipids, especially phospholipids, which contributes to the formation of membranes. So overall, the function of endoplasmic reticulum is to synthesize proteins, lipids, to modify them, and to transport them in the cell, inside the cell, where they are needed, where these substances are needed. So next is, they are also the skeleton of the cell. They provide strength to the cell. Then is Golgi bodies. Golgi apparatus, it is present near the nucleus. Golgi apparatus, they are also membranous dumbbell-shaped structure. There are 2 to 4 in animal cell, but there are 2 to millions in the plant cells. There are much more in the plant cells. The Golgi bodies, their function is to store, to modify, to pack, and to transport the material inside the cell wherever it is needed. Which material? Which is synthesized by the endoplasmic reticulum. That is, some proteins, they are converted into enzymes, some lipids, they are converted into hormones. So they are modified and then they are transported, they are packed and transported to the site where they are needed. How they are transported? The substances which are stored here, when it is to be transported, it will pinch off from it here, and in the form of Golgi vesicle, it will be transported to the site where it is needed. Then next we come to, here we can show, these are the endoplasmic reticulum. Upper one, rough endoplasmic reticulum, and lower one is the smooth endoplasmic reticulum. And this is the Golgi apparatus. These are dumbbell-shaped structure having the Golgi vesicle associated with it. Next structure is, next organelle is mitochondria. These oval-shaped structure which are scattered here and there in the cytoplasm, they are called as this orange color These are called as the mitochondria. Mitochondria, it is double layer structure. Outer membrane is smooth, inner membrane is folded, and these folds are called as the cristae, and the spaces between the folds are called as the crystal space. Inside the mitochondria, it is filled with fluid which is called as the stroma. Mitochondria, they have their own DNA, they have their own ribosomes. So mitochondria synthesizes protein itself. So mitochondria, they are called as the powerhouse of the cell. They produce ATP from the ADP, that is adenosine triphosphate, which is called as the energy currency of the cell. This ATP is used in the cell for various processes, that is synthesis, breakdown, etc., digestion, and also the mechanical movements of the organism. These mitochondria, they are much more in the animal cells but fewer in the plants. Then next we come to the lysosomes. These small spherical structures, they are called as the lysosomes. They may be spherical, oval, or irregular shape. Lysosomes, they contain the digestive enzymes which are acidic in nature. These digestive enzymes, they help to break down the larger particles into smaller particles. If from the outside some food particles is taken, these lysosomes will digest that food particle, break down into smaller particle by its enzymes. So it helps in the synthesis, it helps in the digestion, it helps in the providing the energy. So lysosome, they are also excretory in nature. Is some food, along with food particles, some other bacteria, harmful bacteria, they are taken inside. Then what will happen? Again lysosome will break down and it will release digestive enzyme and will kill the bacteria and digest it. So it is also protector of the cell. It protects the cell from the foreign bodies. If some subcellular organelles, they are old, they are worn out, lysosome will digest those organelles as well and remove it from the cell. So they are excretory in function. So similarly, if the cell itself, it becomes old or it is worn out due to some metabolism, again the lysosome will release its digestive enzyme and will digest its own cell. So they are also called as the suicidal bags. So what is their function? Lysosomes: digestion, excretion, protection, cleaning, right? In addition to these structures, there are present small vesicles which store the lipids, phospholipids, proteins, sugars, etc. And some vacuoles are present which contain the sap-like structure. In addition to these, there are small filaments or tubule-like structure present in the cytoplasm which form the cytoskeleton of the cytoplasm. Cytoskeleton of the cell. They keep the cell stretched. These are called as the microtubules and microfilaments. One more thing, near to the endoplasmic reticulum there are present centrioles. Centrioles are 2 in number. They are 8 folded like structures. These centrioles, during cell division, they duplicate, they replicate, and help in the formation of ester formation and spindle formation during the cell division. So this is all about the cytoplasmic subcellular organelles which are present in the cytoplasm. Then the major part, near to the center is present a round structure bounded by the membrane which is called as the nucleus. This This is nucleus. Nucleus, which I have drawn purple color, actually they are not colored inside the cell. For the sake of convenience, I have given them the colors. This nucleus has three parts. Upper outer layer, that is bilayer. It is porous, which is called as a nuclear membrane. In the nucleus inside, there is present this dense round structure, which is called as the nucleolus. Inside the nucleus, there is present network of thread-like structure, which is called as the chromatin material. You can see this nucleus. This is the nuclear membrane. This one is the nucleolus, and these are the nuclear pore. The nuclear membrane has pores. It is porous, so that substances can pass inside and outside the nucleus. This chromatin material, during the cell division, it is condensed into chromosomes, V-shaped like structure, U-shaped like structure, which are called as the chromosomes. Chromosomes, they are formed of DNA and associated with protein. DNA deoxyribonucleotides, deoxyribonucleic acids associated with the protein. Some of the DNA associated with the protein, the segment forms the gene. So they transmit the genetic characters of the parents to the offspring. These genes, they are responsible to control all the activities of a cell, just like cell growth, cell reproduction, and synthesis of some material. They control. And chromosomes, they determine the sex of the offspring. Nucleolus, it is the site of ribosome synthesis. Ribosomes are synthesized in the nucleolus. This is all about cell structure. Then single nucleus is present in the cell. In some cases, what happens, during cell division, the cell becomes multinucleated. And in some cases, the nucleus is lost, as in case of RBC of the human being. So, that's all about the animal cell structure. And in the next video, we'll describe the plant cell structure, which has few differences with the animal cell structure. I think you must have followed it. Like it, subscribe it, and share it. Thank you. Bye.